does it take to move from decoding neurons in the lab to decoding regulations in biotech boardrooms? How do you bridge the gap between neuroscience and global innovation? And why does that gap even exist? What does it mean to have an atypical career in neuroscience? And how can legislation, startups, and international expansion fit in one career? Welcome to Neurocareers Doing the Impossible, the podcast where we explore the extraordinary paths of those redefining what's possible at the intersection of neuroscience, technology, and human potential. In today's episode, we're joined by Dr. Amanda Wiggins, CEO of the CGP Lab and a true pioneer at the crossroads of science, business, and biotech innovation. With a PhD in neuroscience and career that spans from shaping New Zealand's human tissue bill to spearheading regulatory frameworks for stem cell therapies and xenotransplantation, Amanda's journey is remarkable. She's launched brands across global markets, led biotech ventures through regulatory labyrinths, and now heads the CGP Lab, where groundbreaking work in cycling Glycine-Proline is redefining the future of neurological health. Welcome, Amanda. It's a great pleasure to have you on our podcast. Can you please introduce yourself and let our listeners know where you are joining us from what part of the world?
Thank you for that very kind introduction, Melina. I really appreciate that. I'm Dr. Amanda Wiggins, and I'm coming to you from sunny Nelson in New Zealand. It is 8 a.m. I've had my first coffee, and I'm really, really looking forward to this conversation.
Thank you very much, Amanda. And good morning to you from Florida. We have 4 p.m. already here. And thank you so much for this possibility to explore neuroscience career in fairly non-traditional way. And we will talk about that more. But now that you mentioned New Zealand, and in our conversation, you also mentioned that there are not as many people living in New Zealand. Yes, about 5 million, you mentioned. So what do we know about New Zealand and the development in the neuroscience domain? Are there any institutions that are working on specific neurotechnologies, maybe a specific neuroscience product? Maybe you can just give us a brief overview of neuroscience, neurotech, and in New Zealand.
Oh, I would love to. So New Zealand has 2 universities that offer neuroscience degrees. Otago University, which is our oldest university and I think our oldest medical school. So, often with medical schools, you get those associated sort of medical, biomedical type degrees. So Otago University, which is down south in New Zealand, and that's where I did my undergrad honours degree. We also have Auckland University, which has also got a medical school, our second medical school, and they offer a neuroscience program as well. Both of them are very, very good universities. I think in terms of international rankings, Auckland University is or used to be the only one in New Zealand that's kind of ranked in the top sort of 100 internationally. There's been some quite notable neuroscience techs that have spun out of the universities. The one that's top of mind for me is a drug that's been approved by the FDA just a few years ago, late 2023, I think it was, called Trofinetide. So this was developed up at the University of Auckland, and it was approved by the FDA for the treatment of a rare disease called Rett syndrome. So the reason I know quite a lot about that is that our chief science officer at the company where I'm the CEO at the moment, the CGP Lab, was part of the team that worked on the preclinical development of that molecule. And it's a synthetic analogue of cyclic Glycine-Proline, CGP, which is the natural version, is what we are working on at the moment. And that's a really interesting drug. It works by regulating the hormone IGF-1. And, that's been really quite widely celebrated here in New Zealand. It's not often that a drug candidate comes out of a university here and makes it all the way to FDA approval. As you know, that is a massive journey. And there's a lot of failures along the way. So that's a real success for New Zealand. The company that owns that IP now and is developing it is based in Australia, and that company's listed on the Australian Stock Exchange. It's doing really well.
Yeah, that's pretty impressive already from the very beginning. Thank you so much, Amanda. Now, can you bring us a little bit back into your past and share with our listeners how did you get on this path into neuroscience? When you were growing, if you remember, whom did you want to become? Was it anything related to biology, psychology, neuroscience? How did it all develop for you?
Great question. Look, you know what you like in your late teens, you tend to be quite idealistic. When I was at high school, I wasn't particularly engaged at high school, actually, with academic work until I got to the last couple of years of my high school. And I happened to have two really, really incredible inspirational science teachers, and also a tutor who was 1 year older than me at school, so also a school student, who they just really inspired a love of science, generally at that time. And I didn't have a grand plan for my life at that point in time. But what I did have was the Otago University handbook of all the courses that they offered. So, I just was flicking through that, reading all the different courses and degrees you could do from, marketing and business, which I was so disinterested in at the time, and came across this neuroscience degree. And it really, really struck me. And I think in my youth and naivety, I thought, gosh, you can really start to understand the seat of human consciousness. What does it mean to be me? What does it mean to be human? And I think at that age and stage in life, you do start to get kind of interested in those types of questions. I was reading really interesting scientific books, including one written by Albert Einstein called Ideas and Opinions, which was a game changer for me. And I just really wanted to dive into broadly, what does it mean to be human? And thought that perhaps neuroscience could give the answer. So I enrolled in the degree, I got a place, off I went, waved my parents goodbye. I turned up down at Otago University, and I enrolled in the 3-year Bachelor of Science degree, and towards the end of that third year decided to go back and do an honours year. And I did that honours year in the pharmacology department at Otago. My thesis was all around the neurodegeneration that occurs with MDMA consumption, which is now known as, ecstasy. It's a street drug. Back then, that was a long time ago. It wasn't present in New Zealand really as a recreational drug at the time. So it was a pretty interesting topic to work on, looking at the effect on the redox state of the brain, essentially, after giving mice that drug.
Yes, that is very impressive. And did you find an answer to those questions? What does it mean to be me? And what does it mean to be a human over those years? Or it came later?
I think, I take a fairly reductive view on that. And it might not be that popular with some of your listeners, but, I think I do tend to be very scientific when it comes to that question. What does it mean to be me? I think of the brain as just being this big mass of electroconductivity. We've got all these neurons, billions of neurons talking to each other through chemicals, neurotransmitters, which translate into electricity, essentially. So perhaps I'm just a bit reductionist on it. We're just a bunch of chemicals and electricity in our brain, and that's what makes us who we are. Obviously, it's a lot more complicated than that, but that's my simplistic view on it.
Yes. And this is the view that you develop over those years of studies. Yes. So I absolutely relate to that. So what happened next? You did your thesis on pharmacology in general, yes? And then what happened after that?
So at that stage, I needed a little break because it's quite intense, and I'd worked very, very hard. I did really well. I got first class honours in my honours year. So I took a little break, and I was just working in cafes and just having a bit of downtime. But a good friend of mine from that neuroscience degree had moved off to Australia, Melbourne, and he was contacting me saying, it's fantastic at the University of Melbourne. The neuroscience department is amazing. You should come over here. So I did. I packed up my life into a single backpack. I didn't have much money. I remember my dad dropping me off at the airport to take my flight to Australia, and he said, how much money have you got, Amanda? And I said, I think I've got about $800. He said, oh, well, let me give you some more money. That doesn't sound like enough. So anyway, I landed over there with a few $1,000s and a backpack, which, wow, gosh, what a time of life to do that. And yeah, I spent a couple of months talking to various professors at the University of Melbourne to try to scope up a PhD and what would that look like. And I connected with Andrew Gundlach, who ended up being my PhD supervisor. And yeah, together we put in an application for funding, and I got a full PhD scholarship from the University of Melbourne, which, was incredible, really, to think back at the time. I don't think life was as competitive back then, perhaps, as it is now. But the thing that got me that scholarship really was how well I did in my honours thesis. So, because I got first class honours, and that was a pivotal moment. I think in life you don't sometimes realise where the pivotal moments are, because if I hadn't got that scholarship, I probably wouldn't have done a PhD. I would have done something else. And yeah, so I enrolled in the PhD there, and that took 4 years. Yeah, it was about 3.5 years of doing the work and about 6 months of writing it up.
Yes, you mentioned University of Melbourne. Actually, I don't know if you know, but we have Melbourne here in Florida.
Oh.
It's a little city, yes, on the coast of the Atlantic Ocean. So it just reminded me of that place. Yes. How did you choose your topic? You mentioned that you were talking to different professors. What made you choose this particular professor with this particular topic?
Yeah, I think the thing that made me choose to do that topic, to work with that professor, is that, the topic was something that I was already sort of familiar with. I guess the other option would have been going down the very sort of molecular biochemistry route, but I hadn't sort of done a lot of that type of work. And so I kind of wanted to do something that I had already was somewhat familiar with the techniques and what they were studying. And I also really just liked the group and the team, which I think is actually very important. It is, you pour yourself into your PhD like I think nothing that you will ever do again in the future. And so it's important to feel comfortable and supported in those years. Otherwise, it could be a pretty long, hard road. I mean, it is quite a long, hard road anyway. Yeah, so the topic that I chose was around a phenomenon called cortical spreading depression. So this is, I guess the analogy in humans would be similar to a concussion, but we used a rat model where you essentially put a chemical solution on the cerebral cortex one side in rats. And what happens is that causes a little bit of damage, and what ensues is a big burst of electricity on that side, the unilateral side. And following that, there's a prolonged period of reduced cortical activity. And the thinking is that this is a protective time. So the brain recognizes that there's been an incident, and it essentially shuts down communication for a period of time. And in fact, the changes last for around 2 to 4 weeks, the biochemical changes. So yes, the first thing was to learn that technique, which is surgery. So that's obviously not for the faint-hearted. And it took me probably longer than expected to learn that technique because I was quite squeamish with it, I have to say. So, you go through the ethics approval and everything's obviously approved, and it's a very humane procedure. The rats are fully anaesthetized during it and they receive pain relief afterwards. But that was very challenging for me. I don't know if your listeners have much experience in the kind of animal surgery space, but I guess I would just suggest that it's worth thinking really hard about whether that's a road you want to go down or not. Probably in hindsight, I probably would have preferred not to have that aspect. But anyway, it is what it is. So the animals are then allowed to recover and then sacrificed at different periods following the procedure. And then their brains, examined for different biochemical changes with a view to identifying neuroprotective molecules. And I guess one day the hope that one of them may be developed further for a neuroprotective protocol.
Yeah. What was the most challenging part of your PhD? And how did you overcome that challenge? Maybe you can provide a suggestion to those who are planning their PhD or just in the process of doing it.
There's lots of challenges in a PhD because it's so all-consuming. So I guess just take your time to really choose the right topic that you're really genuinely passionate and interested about. And I think doing a PhD is not for someone who's just a bit interested. You've got to be really in, because you can spend a lot of time doing these techniques and procedures and get results that you can't interpret, right? Which can be devastating. It can be months of work and you get to the end of an experiment and you just don't know. You need more experiments. It's sort of, how long is a piece of string? You could just always keep going, right? There's always more to know. I think, honestly, for me, the most challenging thing was the animal surgery. I really struggled with that aspect and, perfusion and formaldehyde and brains. And, to this day, I still sort of doesn't feel great. I don't really like thinking about that aspect. So, if animal surgery is in your PhD and a technique you need to do, just have a really good think about whether that's what you want to do. Sorry, just as a follow-up to what was challenging, I also found the writing up phase really challenging. And that may just be a personality thing if you're a bit extroverted, like I am. So at the University of Melbourne, once you get into the writing up phase, they give you this really gorgeous office, well, they used to back then, in their main student office building, which is a really lovely Gothic architecture. It's just a gorgeous building, which you would love to be in, right? But for someone like me who's very extroverted, I get my energy from talking to other people, being around people. Going into my own office all day, every day for 8 hours was quite challenging from a mental health perspective. And I think I actually got a bit depressed towards the end of that. I really wasn't the best setup for me. I would have been better having just an office in the lab where there's all the noise and, stuff going on and people. But they want you away from all the noise so you can just focus on writing.
Yes, that's true. And thank you for both things that you mentioned. The first one, I agree about the surgery, that I observed some people who were involved in projects with surgery, specifically students. And I know that not all of them can do and feel good about doing that. Some of them, when they try, they're absolutely amazing. And usually they just do all the job for everyone. I met this person, but the others usually, are hesitant or not very comfortable with that. So in that regard, probably for our listeners who are contemplating any position like this, any work like this, it would be good to go and do some practicum. Yes, internship in the lab just to see if it's really theirs. So in that case.
Absolutely. Yeah.
And with the being extroverted and being alone in a lab, in an office, yes, I also absolutely relate to that. And probably for those who were alone at home during COVID, yes, who were not able to come and work with other people, that also was a challenging time, like it was for you to be in that office. I'm happy that we're out of that time. And because you had challenges, yes, like, of course, many students do. What motivated you to still keep going, even in spite of those surgeries that you were not comfortable with, yes, and being alone in the office? It's not like a big team project, yes, that everyone is cheering and doing together. So what helped you to still stay on track and finish your PhD?
Honestly, Milena, it's just the fascination with the brain. So, I mentioned that I'm pretty reductionist when it comes to that sort of question of what makes us who we are. And our brains are just a big bag of neurons doing it. I know it's more nuanced than that. And I think it's just that real fascination. I mean, the brain, it is just incredible, isn't it? How complex it is. And then when you think about something like, at the end of that writing up period, my mood was very low. You think, gosh, this bag of neurons is not firing as it should, really, right? It's just not working. Obviously, there's an imbalance there, and there was. That to me, that basic fascination with how bit of an imbalance in our brain chemicals can severely impact your mood and who you are and your sense of identity. So, yeah, it was that interest, really, that just kept me going. I also wanted to mention, I'm not sure of the age of your listenership, Milena, but this might be a funny one for people to hear. But back when I was doing that PhD, we were doing a lot of, perfusing brains and then doing fluorescent staining for different things, taking a lot of photographs. It was before digital cameras even existed. So I would spend a long, long time taking actual film photos down a microscope. And then you would have to book the darkroom and go and develop your film and print photos. And then you had special techniques for getting those photos into your print PhD thesis. it was really manual, right? And that would be so much easier now. But I think about that, all those many hours in the dark room, but it was kind of fun too. It was, it's real hands-on stuff.
Yes, yeah, that's so funny thinking about how the technology was primitive. Yes, at that point, I also remember doing a part of my master's thesis at the University of Manchester Institute of Technology. And I worked with the evoked potentials, visual evoked potentials, and we needed to actually just draw it from the screen. Yes, so you put like a transparent paper on a screen and you just trace it. That evoked response. Yes, so we even didn't have a printer at that time that can print from that device. So it was very, very, very different. So, yes. Yeah.
Wow. Yeah. So where did you do was it a master's thesis? Was it? Where did you do that?
That was master's. PhD then later was at the University of Helsinki, but it was after 7 years. And yes, we had already, great equipment and whatnot. So, but those initial stages, and probably the lab wasn't the newest one at that time. But yeah, it was challenging. So I understand. Yeah. So Amanda, at which point did you decide that, okay, I'm good with academia and I want to explore something different and maybe more into the regulatory work? Yes, as I understand, you went to more leadership route and regulations, or can you tell us more about that? What happened?
Yeah, so after I finished my PhD, I fell in love with a boy who then later became my husband. He was at that time, that was towards just right at the very end of my PhD, he was waiting on his visa to be approved to go off to a job in Silicon Valley. And I didn't really have a clear plan. I was starting to have a few doubts about my suitability to neuroscience because of all this writing up in the room. And it just wasn't, but anyway, love took me over to San Francisco where I found a really great postdoc. So we lived in San Francisco for 4 years, and I was out at the Veterans Affairs Hospital, which is associated with UCSF. So I was enrolled with UCSF doing my postdoc there. Molecular biology of brain, completely different. We made a knockout mouse. I said right at the start, I don't want to handle the mice. That's not my role. So it was quite a big team working on this project. We had great success knocking out, a transcription factor called BRING3A, which is involved in the sensory nervous system of the face and neuro. So that was the sort of the main discovery there. But yeah, so the switch, so towards the end of that postdoc, I really knew at that point, this is really not for me. So period there where I had spent about 2 weeks in a dark room and using the scanning electron microscope, you go to work, you shut that door and you're in there the whole day. It doesn't align with my personality. So towards the end of in my last year living in San Francisco, I connected with the American Society for Cell Biology, and they have a really interesting, I guess what you would call a lobbying program. I think they call it something fancier, more like government liaison or something like that, where they will get postdocs and PhD students over to Washington, DC to go and talk to senators about research funding. So you have sort of a script, you say who you are and what you're doing. And our main thing was around NSF funding and why we think it should be increased each year in line with inflation so that the purchasing power of labs continues to stay at pace with what they can buy. So that I did a couple of those trips and it really struck me at that point that, gosh, you can still be involved in the science world, but at a different level, at a more strategic level, at the funding level or the policy level. And that was a real breakthrough for me because that's kind of big picture thinking. And it's out there talking to people, which is probably, honestly, much more aligned to my personality. So, yeah, as we were starting to get ready to move back to New Zealand, as my postdoc was coming to end, I started reaching out to people who were back here in New Zealand in what was then called the Ministry of Research, Science and Technology, and saying, hey, I'm coming back. This is who I am. Do you have any work for me? Which they did. And so I landed and basically walked straight into a pretty cool role I think that my role started off as advisor and then I went to sort of senior advisor. I was working in the emerging biotechnologies team, within the Ministry of Research, Science and Technology. And my very first task was to, quote unquote, their words, sort out the human tissue bill, which was subsequently passed into law and has become the Human Tissue Act 2008. And that was quite a big piece of work and fascinating. Still one of the coolest things I've worked on, actually, that was being run by a different government department, the Ministry of Health, who didn't actually have any scientists advising on this piece of legislation. So I took it home. I read it over a couple of nights. It was this big sort of doorstop of a thing. It's very sort of technical legislative language that I wasn't that familiar with that kind of language. So, it did take me a while to get through it. But what I realized quite quickly is this is going to be really problematic for scientists and probably very embarrassing for government as well, because it had outlawed any sale and purchase of human tissue. The purpose of it was to stop the trade in human organs, right, which is a good thing. But it had inadvertently would have outlawed laboratories purchasing things like cell lines or human tissue slices or samples of cancer cell tumours, that type of thing. So it's like, oh gosh, this is really quite a big red flag here. There's a lot of unintended consequences that, because obviously they've had no scientists look over it. So I took it under my arm. I went off to the university that's in Wellington. That's the capital of New Zealand. They have a science department, but not a neuroscience department. So I popped into one of the labs and had a chat to them and said, look, have a look at this. What do you think? And I said, oh God. So I said, can I borrow some of your catalogues, your product catalogues, and take them into my meeting? And so that's what I did. I had a meeting with the Ministry of Health team and this big team of very serious-based people whose job is to actually write legislation. That's what they do. So I sat down. I felt like I was pretty new and young and, and amongst these kind of very senior, serious people. I said, look, I've got a problem here. Look at these catalogs, tissue catalogs, cell line catalogs. I can open this here. I can say, look, I can buy slides of particular tumor types because I want to research what the profile is in that tumor, cellular profile. That this would become illegal under this proposed legislation. Silence. I said, look, and here's another product catalogue, and it's cell cultures. So I had to explain, this is a really big international thing. Purchasing cell cultures and using them in science is really important and done globally, not just in New Zealand. And if it becomes illegal to purchase any form of human tissue, this is going to be outlawed, and every biology lab in New Zealand will be infringing the law. So we've got to make a change here. And so, what ensued was many months of back and forth around how do we carve out allowances for this type of thing, which is very, very different to black market sale of human organs, right? Very different. And so that's what we did. And it was a pretty exciting time. I was working in a high rise, which had a total complete line of sight to our government building, which is called the Beehive. It's shaped like a beehive. And I'd get to work, say, right, Amanda, please get ready to go over and brief the minister at 8:03. He's got a breakfast, meeting, blah, blah, blah. And so off I'd go, trot over there. It was really cool. It was really interesting work and, a good match for exactly what I wanted to do, which was to be out and about putting my science to use in a different way, at a more kind of strategic, high-level way. And, that was passed into law. So that was passed in 2008.
So how did you manage to actually save the possibility for scientists to purchase the tissue and prohibit the use for those who are trying to get it for transplants, yes, and trade it?
Yeah, so we just made a carve out for some really specific uses. So, listing some examples and laboratory use of, academic use of, that type of thing, while still maintaining the overall intent, which was to prevent the sale and purchase of, organs and blood and that type of thing.
So you were at the right place at the right time, yes?
Yeah,
If you weren't there, I don't know, there would be many problems for scientists. Yes?
It's been a real problem, yeah.
That's wonderful. And what I also liked from our conversation, that you proactively contacted people in, was it New Zealand or it was Australia? Where did you return?
New Zealand, back to New Zealand. Yeah.
New Zealand, yes, back to New Zealand, yes. So you already contacted them even before coming back.
I actually I did even a step further than that, Melina. So at that time in New Zealand, there was a huge public, really fiery debate going on about the use of genetic modification. And that's what became what's called a moratorium. It's a technical term, but it basically means we're just going to put a hold. We're not going to allow any genetic modification in New Zealand, but we're not banning it. We're kind of going to keep a watching brief. Moratorium is what they called it. But there'd been protests at Parliament. New Zealand is at that time, very clean green country, and people are passionate about keeping it that way, right? So that was going on back here in New Zealand. I was living in San Francisco, so I saw an opportunity to put an opinion out. I actually wrote an op-ed piece for the main newspaper, the Wellington newspaper, which they published. They paid me something like $285, which felt like quite a lot at the time.
Yeah.
And in that op-ed, I made the argument for why biotechnology can be very beneficial for societies. It can improve, crop yields and reduce pest infestations. And I wrote about, at that time, some of the really cool New Zealand biotech companies that were going quite well on the international stage. And so that helped me get the job because it was such a hot topic. It was just the right time. I came in, no one really knew who I was because I'd lived overseas for so long. Everyone was like, who is this Dr. Amanda Wiggins publishing in the paper? A big paper, half a page. And back in the day when people read newspapers and, physical copies of newspapers were in every lunchroom, especially around government buildings. And so that, I think, that kind of helped my cause there. So I think, if you've got listeners who are contemplating switching into a government role, try to get your name out there, try to write an article or create yourself a bit of a profile with an opinion about something that's important for society and particularly the interface of society with science. Yeah, that's what I would suggest. Get your name out there. Show that you're someone who's thinking about the impact of science on the world, how it can be beneficial, what the risks are, how you would manage those risks. There are risks to genetic modification, actually, and those do need to be addressed and identified. So it's that kind of balancing, the potential for human benefit from innovation, but also managing the risks at the same time. And that's really what regulations are all about.
So did they change at the end of the day? Did they stop the moratorium, or how did the story end?
Oh, unfortunately, it's not like a big end. It's more like a rolling series of changes where they relax the rules somewhat. So now in New Zealand, yes, you can undertake genetic modification trials in the closed environment, but it's a very, very onerous process to get through. There's lots of checks and balances. So yes, we still technically have the moratorium in place, but there's now avenues for experimenting with genetic modification, particularly around field crops and things that have to be contained until you can determine what the impact is.
Oh, so good that you're mentioning that, because we have right now many regulations that people are trying to put in place in terms of ethical use of neurotechnologies, for example, yes, implantation and possible breaches of confidentiality and unethical use, and to whom they can belong, yes, who will be taking care of them if, for example, the company that implanted gets bankrupt and they cannot support the device anymore, what happens then? So there are so many issues that are related to that, and I think people who are trying to get those regulations in place, they can think of you as an example who did so many things in biotech industry, which I think there are many relevant things that they can learn from you and from your experience.
Wow, thank you. Yeah.
Of course. Thank you. Thank you for mentioning that. So how did you feel after you really made your contribution, yes, with this regulation, and where did it bring you next?
Oh, I was elated when that passed into law. It was a significant piece of work and a significant win for the research community as well. And very interesting for me to get out and about talking to lots of researchers around how they use human tissue. So that was really good. I also got involved in another initiative that the government was working on at the time called the Biotechnology Research Roadmap. So I guess here in New Zealand, we have a fairly limited pool of government R&D funding. And so the government tends to like to identify areas where they can get the biggest return. And so biotechnology was big at the time, and the government wanted to put an R&D roadmap in place around funding and where it goes over time. And so I got involved in the project just as it was towards the end of being written, and my job was to go out with another fabulous woman and go and socialise and present this biotechnology roadmap to all the research institutions around New Zealand. No, that was so bad. Researchers don't really like government telling them what they should be researching, right? And they don't really like a top-down approach identifying the most important areas for New Zealand's biotech R&D. They want academic freedom to do the research that they want to do. So some of those meetings, researchers would stand up and yell at us, tell us to get out. They don't want to see it. And it was not great. But yeah, anyway, that piece of work has sort of become a bit famous in New Zealand because it was so controversial. And anyway, unfortunately, fast forward to now, biotech is not really that much of a focus, not as much as it was back then. And, the country continues to be fairly constrained in its ability to fund R&D, like many countries. As a small country, there's only so much government funding available. So, I did that. I worked on a couple of another really interesting piece of regulation I worked on was in New Zealand, when every baby is born, they take a tiny blood prick from the toe and they every baby's blood for metabolic diseases. It's a newborn screening program. And every single one of those blood spot cards is held in a huge warehouse. So there was a refresh going on around the regulatory framework for that. And I came in pretty hard arguing that researchers should have access to those blood spots so that they can conduct research. What a huge database of human blood samples. So that caused a bit of a ruckus as well because, privacy and consent. Consent is a huge issue. And what if you gave your baby's blood spot and you don't want that baby to be part of the research project? So the way we got around that was to introduce a consent process, consent form at the time of the blood spot being taken that just simply explains that this blood spot may also be used for research, and the mother can sign that or choose not to. You can opt out. And also any researchers who wanted to access those blood spots have to go through an ethics committee approval. So again, it's that kind of checks and balances. I mean, obviously there's huge potential to have research conducted on this massive database of every baby ever born in New Zealand, but you've got to have checks and balances, right? People value their privacy. They value their own genetic data. So yeah, the checks and balances, I guess, is the key message there.
Yes. And also what I've noticed from your experience that I think resonates with the experiences that many people have, that sometimes we have successful projects where we feel good about what happened, yes? And that was your initial project. Then with this biotech roadmap, things were not going as nice as you would like them, yes, to happen. And there were people who were, like you said, even screaming or behaving differently. But we can always take the best from all our experiences, learn from them, and use them for our benefit in the future. So, first of all, how did you handle those situations where things were not going as smoothly as probably you would like to? And how did you use that experience in your future career for your own benefit?
It's difficult when someone you're speaking to is very emotional. I guess the learning for me is it's not the right time to try to rationalize with them. Probably the best approach there is just try to empathize. I can understand why you would be upset. Let's take this offline and see if we can address your concerns. I think my takeaway there is never try to go head to head with someone who's really hit up. It's probably not going to be successful. For me, the takeaway there was just, people have big emotions. They're very passionate about research, scientists, and academic freedom is really a big thing, and we need to respect that. I think that was my real takeaway.
Yes, beautiful, beautiful. Not an easy experience, but who said that it should be? Yes. So you learned a lot, and it was an important experience. So what happened next? That's a lot already. Yes, but what was going on?
Well, so I had always had in the back of my mind that I really wanted to get into industry. I wanted to get into the biotech industry. It's quite a small industry in New Zealand, and I kind of started talking about that but externally I started connecting with a few people. So this is after I think I was in that government ministry for about 8 years altogether. And it was still to this day, some of the best work I've ever done, actually. Really very interesting, that interface of society and science. So, but anyway, I started talking around town, jobs in the biotech industry. What can I do? And I was approached by business incubator in Wellington called Creative HQ. They wanted to pilot a program around commercializing science, providing services to scientists to help them get through that very sort of early stage of working out whether their science has got commercial potential. So they offered me a 1-year fixed-term contract to sort of give this a bit of a go, see how it would go. And I was a sort of a program manager, I guess you would call it, and my job was to go out and talk to research scientists about their research and help them identify where there was commercial potential. So we would, my week was really cool. It was really interesting. I was on the road quite a lot, going to meetings, talking to people about their cool stuff they were doing. And we had a framework that we would put their work through. I would often do it, or I'd contract someone in, a sort of a triage, we called it. We'd do we'd spend 40 hours just researching whether this idea had commercial potential or not. What does the competition look like? How do you get such a product to market? Is it feasible from a regulatory perspective? What are the barriers, that type of thing? How unique is it? Is there an IP position that the company could take if there was a company? So it was a kind of a rapid fire triage of a science idea with a commercial lens. It was so great. But unfortunately, we couldn't secure the ongoing funding for that. It was a government funded program. So just a 1-year fixed term contract. And luckily for me, towards the end of that, I connected with a company who was associated sort of with the program, with the business incubator program generally. And they said, we need a regulatory manager. And I said, well, I'm about to come to the end of my contract. This is sounding really good. And I took up the job. So that was a biotech startup. They had a really high-tech way of growing a particular type of algae indoors in a bioincubator, and they were extracting out a high-value nutraceutical called astaxanthin. And I joined them, and I was working on some of the regulatory stuff, but I don't know how much you've had to do with startups, Milena. You end up sort of doing everything. I took on the marketing role, and that was my first foray into the natural health product industry as well.
Yes, and I'm very curious, what attracted you to industry? You mentioned that you always had somewhere on the back of your mind that, yes, you want to have that industry experience. So what was it?
Oh, look, I think it's just that translation of science into real world impact that really interests me. And I'd had that 1-year contract sort of working at that kind of the very earliest stages of that translation journey that science needs to take to get into society. But businesses is where it actually hits the ground, right? That's where you need the right amount of money to create a product and get that product marketed and sold. And that, to me, is really the ultimate. It's like we're doing all this work in dark rooms around different cities of the world, staring down microscopes for countless hours and animal surgeries that don't feel that great. And all of that is so much work. We need to get these results out into benefit for humans, health benefit, societal benefit, environmental benefit, food protection benefit, whatever it is. I'm really passionate about that, getting these findings out into society. Yeah, but it's challenging, right? I mean, taking something from an idea to a product or a service is a very, very challenging journey. And here in New Zealand, the statistics are very clear. 90% of startups fail. They run out of money.
How many? What is the percentage of startups that fail?
90%.
No. Oh.
I know. And the main reason, is that they fail to scale to significant enough revenue and they run out of money. So with, R&D based companies, there's a huge amount of investment required in the actual R&D itself. And then the productization of the R&D, the regulatory approval pathway, depending on what it is, can be incredibly complex and long. And that all takes investment because you for probably the first number of years, you have no revenue until you get a product into market. So that is a very challenging situation. And that's the underlying cause of like the 90% failure rate. Yeah, I smile, but it's hard.
Yeah, it is hard. Do you see people in these companies trying it again next time, maybe with different approach, with different company, with different product, and maybe succeeding? Or how many attempts does it take for a person to go through this process to succeed finally? So what is your observation?
I think, in New Zealand, what we see is a lot of recycling of talent. So as you pointed out, Melina, once you've done it once and failed, you learn the pitfalls, you learn what you would do differently. And we see a lot of that. We see a lot of young people who've had a startup that failed who come back in with another really good idea because that entrepreneurial spirit is really alive and well in them. My other sort of general observation is around scientists leading companies is often not a good personality fit. A lot of scientists are quite introverted. And when you're leading a company, you've got to be quite extroverted. You've got to be out there talking about it a lot, raising capital and promoting what you're trying to do, building a dream, right? So there's a bit of a mismatch of personality types there from what I've seen. So what tends to happen in the journey of a startup is they'll get to a certain point and they'll bring in a CEO who's not the original scientist. And that's what I'm doing now. So I work for a company now. The company's called the CGP Lab. I'm not the founding scientist. They've brought me in as the CEO to drive the growth of the company and take it through to the next stage of development. And it's like this kind of perfect culmination of all these other things I've done, the neuroscience, regulatory, the love of being out there talking to people and whatnot is it's like a perfect role that was almost just created and kind of here I am doing it. It's great. Yeah.
Yes, it was created just for you. Yes, it looks
and that's
like.
how it feels.
Yeah.
Right place, right time, once again.
Yes. I remember that Steve Jobs said in one of his talks, or it was an interview, that there is one point in life when all things fall into place. Yes, so because when we're going through life, we're getting one experience or another experience, and we really don't see how this all can play together. But there is this point, like it happened for you, that everything came, yes, and dark labs, where you were photographing, doing your research and the regulatory and your initial experience in industry, it's all came and provided you with this opportunity to really now showcase all your talents and experience that you acquired.
So I think, I think my career is somewhat atypical because, I've been an academic researcher, I've worked in government, regulatory, and now business. And I think that's not normal to cross so many different spectrum, right, of types of jobs. But for me, it keeps me interested. I'm a person who likes learning new things, and I've been in the business world for 11 years now, and I'm just learning so much. I really wish, I think back to when I was flicking through that handbook of Otago University, looking at the business papers thinking, oh, back then I thought, oh God, boring. I really wish I'd taken a few business papers, of course, to have some formal education in business, but I didn't. And so I'm learning on the job, learning as I go and doing quite a bit of reading as well. That's kind of my career story. And now I'm leading a company. We're commercializing a dipeptide molecule, which is called cyclic Glycine-Proline, or CGP for short. And CGP is really, really interesting because it's a very neuroactive molecule. It crosses the blood-brain barrier and it gets into the brain and it does a similar thing in the body as well, actually, but it regulates the hormone IGF-1. And so it does that by competing with IGF-1 to bind to its binding protein. And when CGP binds to the binding protein, it dislodges IGF-1, where IGF-1 can be more biologically active. And IGF-1 does lots of different things, but that we're most interested in for people who are about 45 age and over is its effect on capillary angiogenesis. So it spurs on the growth and repair of tiny capillaries in the brain. So blood flow and the brain, a great combination, especially as you get over the age of about 45, where we lose 3% of it on volume of blood flow in our brain each year. So yeah, it's very important to look after your blood flow. And it's perhaps not something that people think about much. There's about 30 years of research on CGP, and the research shows that people who have a higher endogenous level of CGP have better cognition later in life. And so there's that kind of real neuroprotective angle. And a couple of years ago, there was also a mouse study published that showed giving CGP was able to improve memory and reduce plaque buildup in these Alzheimer's transgenic mice. So there's a huge potential for CGP, and we're just at the start of that journey. We've got a dietary supplement brand that we sell called CGP Max, and we're now investigating a whole new sort of innovation, which is the ability of CGP to improve peripheral neuropathies as well.
Yeah, that's very impressive. Are you synthesizing it or you're extracting it from some natural sources? How do you do it?
Yeah, so we manufacture it from New Zealand food sources. So we've got a proprietary manufacturing process that combines New Zealand blackcurrant with bovine collagen. So yeah, there's an amino acid profile between those two ingredients of high glycine and proline content. And we put that through a prolonged heating process, which cyclizes the glycine and the proline together into that cyclic dipeptide, and then we standardize it so we know that we're giving a standardized dose. Yeah, so that's taken the company 2 or 3 years to really develop that manufacturing process.
Yes. And what are your future plans? What are your next steps? Where are you heading with this product, or maybe with new products? Yes.
So we've done a pilot stage clinical study with our product, and it was in people who had type 2 diabetes together with peripheral neuropathy, which is where those peripheral nerves start to degenerate and people get a lot of pain and tingling and eventually loss of sensation, usually in their feet, which is really, really problematic because you can have loss of balance. People can damage their feet. They don't know if they've got something like a diabetic ulcer because they can't feel it. So our pilot stage clinical study showed that our CGP supplement is very effective for restoring sensation in the feet. And we're really excited about that because there's no therapeutic available that does that. There's pain medication that's approved, but nothing that actually restores blood flow. So we're very excited about this result. It was just a pilot stage clinical study, so very small numbers and no control group. So we really want to undertake a randomized placebo-controlled study next, and we're raising capital to make that happen.
Yes. What was the most challenging part for you transitioning from regulatory type of work in the government into this entrepreneurial space, the space of startups?
I think it's probably just that feeling of imposter syndrome. I don't come from a business background. I don't have any formal education in business. So I have, probably more in the past, just felt like, I don't belong here, or I don't have the knowledge, or I just feel like a bit of an imposter at times. But that's going away now. I've been in the business world for 11 years now, and I feel more and more, I guess the word is qualified to be here every day. Yeah. And I've also found in my journey across these different types of industries, regulatory business, that generally people are very happy to act as a mentor. I've been fortunate to have some really incredible people in my life professionally who've helped me out, who've advised me, assisted me to understand things that I didn't understand. And I would just recommend to your listeners, just really lean into that. Lean into the people around you who are really experienced. And, there are certain people who are incredibly generous with their knowledge and their time. They'll take the time to have a coffee with you or a follow-up call or whatever. Just really lean into that. Enjoy that. And then when you get to this stage in life, my stage in life, be that person to others. Take the time to have a coffee with a more junior person. Tell them what you see, what they could do differently, or be the mentor. I really appreciate certain people in my life who are very generous with their time and expertise. So,
Yes, thank you for mentioning that. And what word of advice would you give to specifically those people who are right now at this intersection between science and entrepreneurship when they are just starting their journey? What would you tell them?
I would say just do it. Just go hard and don't be afraid of failure. Chances are your startup might fail. The statistics say it might fail. Don't take that personally. Just learn the lesson. I think really pragmatically also, probably don't ever put your house on the line. Just financially, startups, the statistics around startup failure are quite drastic. make sure you protect your own assets. That's a real practical insight.
Yes, that is a good one. And can you recall things in all your career journey that seemed as impossible at the beginning, but you eventually made it possible? And if so, how did you do that?
As I mentioned, I really wanted to get into the business world, but I couldn't see a way. I wasn't at a time in my life where I could go back and do an MBA. I'd had 2 children by that stage. I couldn't see a way through that. But again, it was just talking to people around me, talking about what I wanted to do, the journey I wanted to take. And this perfect opportunity came up that enabled me to start to enter the world of business. And I'm very grateful for that opportunity.
What do you see the current tendencies in the need for biotech product in the neuroscience space? What people are looking for? What is the need? Where do you see the industry is going in the near future?
Oh gosh, I mean, there's so much need out there. I think the thing I've become pretty hyper-focused on dementia prevention. Hyper-focused. There's a lot of other need, obviously, but just for me personally, we've got a couple of family members who've been affected now, and also friends of my age whose parents are affected by dementia. It's just so heartbreaking to see a loved one lose themselves. Yeah, so for me, I'm watching the pace of dementia research, and I see, gosh, there's a lot happening. There's a lot in the prevention space, the diagnostic space, and some traction in the treatment space, but not as much as we need. But I think where we really need to shift the dial there is much, much earlier detection. So we know that the earlier stages of dementia actually could potentially be detected around age 30. And so if we had that tool, what could we do with that? We've also got quite good knowledge around prevention, right? Modifiable risk factors, things like diet, exercise, connection, nutrition, those types of things. What could we do to turn the dial on the rising tide of dementia? That, to me, that's where my thinking is at the moment. What can we do in this space, to help families not have to go through this in the future?
And how do you see career opportunities in the same space of biotech related to neuroscience? Do you see more opportunities in the industry, in the regulatory field? Do you see any changes that are happening between those spaces, like academia, regulatory, and entrepreneurship?
I mean, AI is the next big wave, isn't it? And how AI and the brain, implantables, that type of thing, that's a whole new world. That's probably the next big leap, right? So back when I was working in regulatory, it was just biotech. Genetic engineering was considered a huge deal. That's kind of become normalized now. If we look 50 years into the future, are most people going to have some sort of implantable device that helps them perform better, think differently? What is that? And from a regulatory perspective, how on earth do you manage the potential balance, the potential benefit to society with risks, things like equitable access? If only some people have access to the technology, is that going to increase inequities in society? So, I'd leave that to the next generation to sort that issue out. That seems really complicated to me. But I think at the same time, the kind of the traditional model of pharma development, that hasn't changed much over my lifetime. Is that going to accelerate and be different? I mean, you still need all the preclinical work, the clinical development, the regulatory approval. I can't see that changing much. So, a career in pharma is a good one. That brings, lab discoveries to humans who need it. I'm really interested particularly in that kind of Alzheimer's space, amyloid plaque intervention. So there's a monoclonal antibody against amyloid plaques that's approved in quite a few countries now. It's not very effective, which is really interesting. So that turns the whole amyloid plaque theory of Alzheimer's on its head. Back when I was studying neuroscience, it was thought that plaques and tangles caused Alzheimer's disease. I think the picture that's emerging now is probably it's a consequence of other changes that cause Alzheimer's disease. So to me, that's the frontier I'm most interested in. If I could have my time again in the lab, that's where I would work, I think.
Yeah, that is exciting. And I remember we had an interview with Dr. Kuminobu Sugaya here at UCF, professor at UCF, and he was saying that a long time ago when he was even just starting his academic journey, he was saying that amyloid plaque is not the cause. And now he is just so happy discovering that, he was right. But at that time, not many people really believed in this. And this happens in science. Yes, sometimes we say something and we believe that we are right, but people may not agree with us at all. And after so many years, we get this confirmation. So that also opens another question, I think, and where is that line where we should still believe in ourselves and in our ideas and keep that belief versus giving up, because of opinions of other people. So did you have any experience like this throughout your life where you really had certain idea, but there was no evidence at that point to support it? No, not really.
No, not really. But I can think of another really interesting example. So when I did my undergrad, I remember very distinctly being told that there can be no new neurons in your brain. You go through that, as a baby, there's a pruning that goes on as babies develop in the first couple of years. And then that number of neurons is set for the rest of your life. So if you damage your brain or anything like drug or alcohol abuse that damages neurons, that they can maybe connect with other neurons, but you can't grow, there's no new neurons. That has been completely turned on its head. We now know that there are stem cells in the neuronal stem cells in the brain, and that there is such a thing as adult neurogenesis. New nerve cells can be born. So yeah, we see these innovations, and I think it's cool. I just love watching knowledge evolve and grow.
Yes, and I see that passion in you. So it really shows, yes, that you have it. Thank you so much, Amanda. If our listeners want to learn more about what you do, about this new product, about you, connect with you, what would be the best way to do that?
Oh, that would be great. I would love that. So I'm on LinkedIn. Look me up on LinkedIn, Amanda Wiggins. You can also check out our product website for our supplement range, which is www.cgpmax.com. Those would probably be the best two ways. And I'd love to hear from any listeners. That would be great.
Yes, and we will add this information into our podcast notes so that our listeners would be able to look at this website and also connect with you. So that's wonderful. Now, I truly appreciate this inspiring conversation and your story of a not traditional path, yes, of a neuroscientist, which I think is very important for people to hear about. Yes, that there are different ways to apply your knowledge and in different ways, which you did wonderfully. Maybe there is something that I didn't ask you, but you still want to share with our listeners. That would be a perfect time. Still something.
Oh, I guess my closing thoughts are, Milena, neuroscience is still the best. Just jump in there. And, if it's not working for you, being stuck in a lab, find a way to apply that knowledge in a different way, because, the love of the brain, the passion to understand what makes us think and what makes us who we are, and to solve real world problems like dementia and many, many other brain problems. Just get in there. Your skills and knowledge are hugely valuable across different domains. So, yeah, I guess if you're not quite suited to lab work, don't let that put you off. Find a different way to apply it. That would be my key take home.
Thank you so much, Amanda. It's been a great pleasure. I and all our listeners are wishing you all the best in your work, and I'm sure you will make a huge impact and on people's health moving forward. Thank you.
Thank you so much for having me. It's been a real pleasure, Milena. Take care.
Dear Neurocareers podcast listeners, thank you for joining me and my incredible guests on this exciting journey into careers in neuroscience and neurotechnologies. I hope you've been inspired by the stories of those turning groundbreaking ideas into impactful realities. If you are looking for more guidance on advancing your neurocareer, book a free consultation with me, Dr. K, your podcast host and neurocareers coach at the Institute of Neuroapproaches, the only career service dedicated specifically to the needs of professionals in neurotech. Let's take the next step toward your neurocareer success together. In addition to free consultations, the Institute of Neuroapproaches offers a variety of services designed to help you thrive in neurotech. First, professional development planning. We help you create a tailored career development plan, identify knowledge gaps, and equip you with the tools needed to achieve your goals in neuroscience and neurotechnologies. Second, resume and cover letter review. Get expert feedback on how to craft documents that are specifically designed for job opportunities in the neurotech field. Third, interview preparation. Sharpen your interview skills through mock interviews and targeted feedback, including soft skills, hard skills in neurotech, BCIs, e.g., MEG, live coding, and presentation preparation. 4. Networking and job search strategy. Learn how to effectively network and uncover job opportunities while standing out in the competitive neurotech job market. 5. Negotiation and salary advice. Receive expert guidance on negotiating salaries and benefits and determining a fair salary range for your desired position. All this and more can be found at www.neuroapproaches.org. Again, www.neuroapproaches.org. So, what are you waiting for? Let's navigate the path to success in the world of neurocareers together and turn the impossible into possible. We're looking forward to working with you.